It's my pleasure to introduce Dr. Devin Koestler, a professor and associate director of research operations in the Department of Biostatistics at the University of Kansas Medical Center. Dr. Koestler received his PhD in biostatistics from Brown University and completed his postdoctoral training in the Quantitative Biomedical Science program at Dartmouth College. His primary research involves the development and application of statistical methodologies for high-throughput omics data, with an emphasis on array-based DNA methylation. His methodological interests include statistical genomics, computational statistics, machine learning, artificial intelligence, and finite mixture models. In addition to his methodological interests, he also has a deep interest in epigenetics and molecular epidemiology. Many of Dr. Koestler's collaborations involve large-scale epidemiological studies for studying DNA methylation and its relation to environmental and lifestyle exposures, as well as the role of methylation in disease susceptibility and prognosis. Currently, Dr. Koestler also served as the Assistant Director for Shared Resources for the University of Kansas Cancer Center. He is also a co-director of Biostatistics and Informatics Shared Resource and the director of the Quantitative Omics Core. And today we are lucky that he will talk about DNA methylation-based deconvolution. So without further ado, Dr. Koestler.
Thank you. That was a wonderful introduction. Thank you, Anna. Really appreciate the opportunity to be here with you this morning to chat about some of my work in the area of deconvolution for DNA methylation data. I recognize that there are a lot of big words to unpack in the title of my presentation, and I hope to make this very high level and accessible to a broad audience. So there are some formulas, naturally as a statistician, there's some formulas in the slides, but I've tried to keep it very high level, pictures, analogies. And so when I do have formulas, I'll make sure to slow down and really kind of try to unpack those with you. So Anna did a great job with her introduction. Couple of the roles and responsibilities that I have here at the medical center. So I serve as the Associate Director of Research Operations in the department that I'm in, which is Biostatistics and Data Science. I am the Assistant Director for Shared Resources for the KU Cancer Center. I co-direct our Biostatistics and Informatics Shared Resource. I also serve as the Site Director for our Data Science Core for our Kansas INBRE, and I direct the Quantitative Omics Core that supports the Kansas Institute for Precision Medicine. So I wear a lot of different hats on campus, but one thing that all of these different roles kind of have in common is it gives me the experience of collaborating with a lot of different types of scientists on a lot of different types of projects. So I get to work with basic scientists, translational researchers, and clinicians. And the really neat thing about that is that has given me the opportunity to kind of fuel my own methodological research. So when thinking about what to give a talk on today, some of the ongoing work I'm doing is in batch effect correction for omics data. I'm also doing some work with analyzing wearable data, so continuous glucose monitoring data. But I thought a good topic to focus on would be kind of presenting a little bit about the history and chronology of one thing that's been a staple of my research going back all the way to when I was a graduate student, and that's been DNA methylation, and particularly deconvolution in the context of DNA methylation. So that's what we're going to focus on today. So this is a little bit of a silly example. What in the world does deconvolution have anything to do with smoothies? But before we get into this example, let me just give you a definition of deconvolution to start. So in a statistical context, deconvolution refers to the process of recovering true signal or distribution when the data we actually observed have been blurred or mixed by some known or unknown process. So the concept of deconvolution, I think, can be really well understood by considering smoothies. So what are smoothies? And smoothies are really just a blend or mixture of different types of fruits that are combined in different types of amounts or proportions. So the proportions of the different fruits that are being used to make the smoothie is going to impact the way that we receive that smoothie. So it's going to impact the way it tastes. It may impact the texture of the smoothie. It may even impact the color of the smoothie. So here's a few examples where if we mix strawberries, bananas, and blueberries in equal proportions, we might get a smoothie that has like a brownish color to it. If we up the proportion of strawberries in our smoothie, then we're going to get a smoothie, a mixture that's a little bit more red because strawberries are red. And if we up the proportion of blueberries, we're going to get a smoothie that's a little bit more blue. So the concept of deconvolution is if I gave you the smoothie and I just told you that there were strawberries, bananas, and blueberries, but I didn't tell you the fraction of each one of those fruits in the smoothie, deconvolution amounts to trying to figure out what the proportions are, just based on the color, the texture, the taste of the smoothie. So we can play this same game with blood. So knowing what types of cell types are present in a given mixture, we can try to determine the proportion that each one of these cell types represents in the mixture. So there are a couple of things that we need to know. First of all, we need to know what cell types are actually present in the mixture. So if we're talking about whole blood, we might be referring to neutrophils, B cells, different types of CD4 and CD8 T cells, NK cells. We also have the assumption that the sum of the proportions of each one of the cell types that we believe to be present in the mixture equals 1. So mathematically, what we're doing when we talk about deconvolution is we're estimating the proportions, these W's, of each one of the cell types that we believe to be present in the mixture. Now, in the same way that for our smoothie example, there are different characteristics of the fruits that help us determine their proportion in the mixture, things like their taste, their texture, or even their color. We can play the same game with cells. There's different aspects about cells that allow us to determine the fingerprint, if you will, to figure out how to deconvolve a mixture. So we can consider gene expression data, DNA methylation data, proteins on the surface of the cell, the morphology of the cell. What we're going to be considering today is epigenomics. So the DNA methylation fingerprint or signature of the cells and using that to deconvolve. So because our story centers around epigenome and DNA methylation, let's start by kind of defining what we mean by these things. So all cells in our body, and they're about somewhere in the order of 200, effectively have the same DNA, but we know can exhibit a variety of different functions and phenotypes. Now, part of what gives rise to the different phenotypes we have in the variety of cells is epigenomics. So when we talk about epigenetics, we're talking about things, the prefix epi means above or beyond. And so epigenetics are things that are above and beyond genetics. And so I think that the relationship between epigenetics and genetics can be really well described by thinking of a piece of music. So in a piece of music, we know that it can sound completely different if played by two different orchestras. So for instance, Mozart's Turkish March, if you were to hear it from the Boston Philharmonic versus, say, a high school orchestra, it would likely sound very different. So genetics are like the actual musical notes, the piece of music that is in front of the musician, whereas epigenetics might refer to the way in which that music is played or expressed. So there are a variety of different epigenetic mechanisms, as we know. DNA methylation is one of them, but there are also histone modifications, chromatin remodeling, microRNAs. When we talk about DNA methylation, it's the process by which methyl groups, or CH3, are added to the DNA, and methylation occurs almost exclusively in the context of CpG dinucleotides. And so while these CpGs are generally underrepresented throughout the genome, they tend to cluster in CpG-dense regions that we refer to as CpG islands, and these are often located near the gene promoter region. About 70% of human genes initiate transcription from these so-called CpG islands. Now, what's important to keep in mind about DNA methylation is that it has the potential to control gene expression, and aberrant patterns of methylation result in the silencing can result in the silencing of genes. And this can be particularly important for disease development if we are, for instance, silencing a tumor suppressor gene. That would obviously not be a good thing, and that could open the floodgates for disease development. So there are a variety of different ways of quantifying DNA methylation. There's sequencing-based approaches. There's also arrays. In the types of studies that I collaborate on, which are typically large-scale epidemiologic studies, the arrays provide a really nice balance of cost, throughput, and coverage. And when we assay data through, say, the Illumina arrays, we get what's called a beta value for each one of the CPGs that are being profiled on the array. So the beta value is defined as follows. It is simply just the ratio of the methylated signal intensity for a given CPG J divided by the methylated signal intensity plus the unmethylated signal intensity for that same CPG. Now, based on the way the beta value is calculated, it's bounded below by 0 and above by 1. So it takes on a value somewhere between 0 and 1. And these beta values can be loosely interpreted as the fraction of methylated cells at that specific position J. So for a given sample where we've profiled methylation, say using the Illumina array, the data that we have, if you think about it visually, is something like this. So what we have here are the 22 autosomes. Each one of these little lollipops would be a CPG site somewhere in the DNA sequence. And the color of the lollipop reflects the beta value or the extent of methylation at that specific CPG. Okay? So we have data like this for all of the samples that we've arrayed. And so not surprisingly, one thing that we might want to do with such data, let's say we've profiled methylation for 5 patients that have cancer. And so we've extracted or biopsied a piece of the tumor and we've profiled methylation. Let's say we also take either adjacent normal tissue from the same individuals, or we have cancer-free subjects that we've collected tissue from and profiled methylation. So we get data that looks like this. Our rows are our different samples, our columns are our different CPGs, and for the latest arrays that we use for assessing methylation, we're looking at somewhere between 800,000 to 900,000 CPG sites that are being profiled in the genome. So this CPG site right here might be a CPG of interest. We can see that it tends to have lower methylation values in the tumor sample as compared to the normal. We would call this particular CPG a differentially methylated position. All right, so hopefully this gives us a little bit of an idea and a starting ground for understanding epigenetics, DNA methylation, and kind of how we assess methylation. So the story of deconvolution has its beginnings in around 2009. So around this time, I guess leading up to 2009, a lot of the studies of DNA methylation and cancer were based on interrogating DNA methylation in the tissue of origin, so tumor tissue, for example. So around 2009, there was a growing interest in profiling methylation in blood and trying to understand patterns of methylation in this tissue type, how it varied between subjects that had cancer and subjects that didn't. There's, I guess, 2 main reasons why blood was of interest here. One is its easy accessibility, the fact that it can be collected using relatively non-invasive procedures. And the other aspect, on more of a biological note, is that there's an immune component to cancer. And so assessing patterns of methylation in blood could provide insight into this process. So what these early studies showed, some of them I had the good fortune of being part of when I was a graduate student and postdoc, is that there were striking patterns that differed between cancer cases and controls in blood. However, there was a question that remained after all of these studies, which was what was driving these associations. So to begin to answer this question, there are key features of DNA methylation and the way in which it's assessed that we need to consider. So the first is the fact that DNA methylation is tissue specific. That is, different tissue types have their own distinct methylation signature. So this is a figure from a paper that was published by a colleague of mine, Brock Christensen, and it was one of the first to empirically demonstrate this. So even though this paper is quite old, going back to 2009, what Brock did was they collected tissue types from a variety of different anatomical locations. They profiled DNA methylation using one of the very early arrays called the Golden Gate array, and then looked at the pattern of methylation and how it varied across the different tissue types. And one of the things that we see in this heat map that's based on a hierarchical clustering of all of the samples is that the samples do indeed cluster on the basis of the tissue type. So we have 3 main clusters that are being shown here. These, the orange and the red, is a cluster that consists entirely of blood samples. We have blood samples from infants, we have blood samples from adults. They cluster together. This cluster all the way on the end, this purple one, are placenta samples. And then this white cluster here is just kind of a mishmash of all different types of other tissue types: brain, head and neck, small intestine, lung, pleura, kidney, and bladder. So we see here that different tissue types have their own distinct pattern of methylation. And if we dig in to a specific type of tissue, let's say different white blood cell types, we see the very same thing. So a few years after Brock published his study, one of our collaborators, Andy Hausman, published a study where he looked at patterns of methylation in different leukocyte subtypes. So here are 5 different leukocyte components of whole blood. Same type of idea here, hierarchical clustering of the different cell types on the basis of their methylation pattern. And we see that the cell types cluster out very neatly. So different patterns of methylation even across different white blood cell types. So why is this a problem? Well, if our goal is to understand the relationship between DNA methylation and, say, cancer or some other type of disease, what we have here is a potential for confounding due to differences in the underlying cell distribution. So the idea here is when we have, let's say, subjects with cancer and subjects without cancer, we would expect the immune composition to be different between those 2 different types of subjects. So we have a relationship between the underlying cell distribution and the phenotype or disease that we're studying. Also, as I just showed you, we know that different cells, different tissues, exhibit different patterns of methylation. So we have a relationship here. So what we have is a classic case of confounding, and that can distort our interpretation of any differences that we see or relationships that we see between methylation and cancer. So to go back to our smoothie example, let's suppose that we want to compare the taste of 2 different smoothies. All right. We have a smoothie that was made at Jamba Juice. We have a smoothie that was made at Smoothie King. And we find that the smoothies do taste different. All right. So there are 3 different possibilities for why the smoothie might taste different. The first possibility is that the proportion of the different fruits vary between the 2 different smoothies. So WJJ and WSK, let's say that they represent respectively the proportion of the different fruits, strawberry, banana, blueberry, in the Jamba Juice smoothie and in the Smoothie King smoothie. So if these 2 smoothies taste different, it could be because the proportions of the fruits that were used to make the smoothie vary. So that's one possibility. The other possibility is that the taste profile of the fruits is different between JJ and SK, so between Jamba Juice and Smoothie King. So what do we mean by that? So even though the proportions of the fruits may be totally identical, it could be the case that the strawberries that were used to make the Jamba Juice were grown in California, whereas the strawberries used to make the Smoothie King smoothie were grown somewhere else with different soil conditions, different weather conditions, and so that leads to a different taste profile of the fruit. So even though their proportions are identical, the actual way the fruit tastes is different, and so it makes the smoothie taste different. The third possibility is that we have some combination of 1 and 2. So we encounter the very same thing with blood cell types and DNA methylation. One possibility why we might be seeing different patterns of methylation between our cancer cases and controls is because the proportion of the different immune cell types differ between the cases and controls. Item 2 is that we might actually have bona fide differences in CpG-specific methylation between the cases and controls. So that's a possibility. And then possibility 3 is that we have some combination of 1 and 2. So to begin to understand the role that different immune composition might have in some of the early results in 2009, 2010, where we had studies that found differences in methylation in the blood of cancer cases and controls, there are a couple of studies that were published in 2012 that shed some light on that. So the first was published when I was a postdoc in cancer epidemiology, biomarkers, and prevention. And basically what this study involved doing was taking leukocyte-specific data. So we actually had collected a data set where we purified and isolated different types of leukocyte subtypes. So NK cells, B cells, different types of T cells, and then of the myeloid lineage, granulocytes and monocytes. And we profiled those different cell types for methylation using the Illumina 27K array. So we were able to identify differentially methylated sites between all of these different cell types. And the question that we asked is, if we take those CPGs that we know differentiate the different cell types, if we take those CPGs and we look at them in these different cancer case control studies where they have methylation in blood, do those same sites that differentiate the white blood cell types also differentiate the cancer cases and controls? So to answer this question, we used 3 different data sets that were previously published at the time. We used a head and neck data set that consisted of 92 cases and 92 controls. We used an ovarian cancer data set that had 266 cases, 131 of which were pre-treatment, 274 controls, and then we used a bladder cancer data set of 223 cases and 237 controls. And so what we did was we identified the top 50 CPGs that differentiate different white blood cell types. And I'm showing those 50 CPGs, showing a portion of those 50 CPGs here on a heat map. We can see that these CPGs do indeed exhibit distinct patterns of methylation across the different cell types. And as I said, we asked the question, do those same CPGs differentiate the cancer cases and controls in these 3 different studies? So what I'm showing here on the bottom is a plot whose y-axis is the minus log 10 p-value from a statistical test of differential methylation between cases and controls. So the higher the point is on this plot, the smaller the p-value was from that statistical test. So what we saw was that in the ovarian cancer data set, 47 out of the 50 CPGs that differentiate white blood cell types also differentiate the cancer cases from controls. 48 out of 50 significantly differentiated the head and neck cases from controls. In the bladder cancer data set, 8 out of 50 differentiated the cases from controls. So much less, but actually this isn't entirely unexpected. When we dug back into the bladder cancer data set, what we found was that they were predominantly non-muscle invasive bladder cancer cases. And at the time of blood draw, most of them had already been treated with BCG, which is an immunotherapy. So it's not entirely unsurprising that there were a fewer number of CPGs that were found to be different between the cases and controls in the bladder cancer data set. But nevertheless, what we see from this study is that a lot of the associations that were being reported in these early cancer studies were probably being driven by differences in immune composition and maybe not so much bona fide differences in CPG-specific methylation. So the first and I would say clearest demonstration of the ramifications of failing to adjust for cell composition in blood-based DNA methylation comparisons, where we have diseased and non-diseased, was really demonstrated by Liu et al. In 2012. But what I'm showing you here are some results of my own. So what we see in the top part of this slide is what happens when we compare ovarian cancer cases and controls in an unadjusted analysis. So what I mean by unadjusted is we're not adjusting at all for immune composition or cell distribution across any of the samples that are being used for this analysis. So in this analysis, we find that there are a strikingly high number of CPGs that are differentially methylated between the ovarian cancer cases and controls. And when we look at a volcano plot where we have the effect size in the x-axis and minus log 10 Q value on the y-axis, we see that of the 27,000 CPGs that were tested, a lot of them withstand adjustment for multiple testing and exhibit statistically significant differences. However, if we actually adjust for cell composition in our analysis, so we throw in an additional term here as we would adjusting for any other confounder, we see an attenuated effect. So many of the CPGs that were previously found to be differentially methylated between the cases and controls are no longer differentially methylated. Again, because they were merely markers or fingerprints of different white blood cell types. And when we did an unadjusted analysis, that was what was being picked up. So going back to our directed acyclic graph. So why is this a problem? Well, it's been noted by some of our work, and it's been noted by others, that heterogeneity in white blood cells has the potential to confound DNA methylation measurements. So this is a really important thing that we want to account for in our analysis. So many epidemiologic studies are poised to pursue EWAs, and they needed a solution. So there are a couple different ways that we can approach this. The way that we would handle any other confounder is if we can measure it, we can include it in our statistical models and adjust for it. So in the context of blood-based methylation, one option would be if we were to have done a complete blood cell count with differential in the same exact sample that was used for methylation profiling, we could take that data and adjust for variation in the immune composition in our statistical models, but that has some limitations. For starters, it's going to require fresh samples, which might not be logistically possible given our study. The secondly is CBC with diff lacks granularity. It only resolves 5 cell types and doesn't even resolve different types of lymphocytic cell types where there could be really interesting patterns. So that has limitations. Another solution would be to do flow cytometry on the same blood sample that's used for methylation profiling. That also requires fresh samples and is an additional cost to our study. A lot of the epidemiologic studies that I've worked on use frozen blood samples that have been, frozen and collected. We couldn't go back and do flow cytometry on those samples. So flow is totally out of the question anyways for those types of studies. So how can we circumvent this issue? And this gets back to one of the key observations that we noted a few slides ago on DNA methylation, which is its tissue specificity. So one thing that we can do to address the potential for confounding due to cell heterogeneity is to actually use cell-specific methylation data to estimate the proportions of different cell types present in our mixture. So the data that we actually observe is represented by Y. This is our whole blood DNA methylation. And what we believe is that that whole blood methylation signature is a weighted mixture of the signal that's coming from the 3 different cell types that underlie the mixture. The proportions of each one of those cell types represented in the mixture are given by W1, W2, and W3. So our goal in deconvolution is to estimate those Ws. And so we have a very simple regression problem here. We have that Y is equal to S times W plus some error. Our goal mathematically is to estimate the W. We have observed S, we have observed Y. We want to estimate the W, but where this differs from a typical regression problem is that we have constraints on the Ws. Because the Ws represent proportions, those proportions must sum to 1. And because they're proportions, they must be between 0 and 1. So we can estimate the Ws using a mathematical procedure called constrained projection or quadratic programming. And this idea was first described in Andy Hausman's landmark paper, DNA Methylation Arrays as Surrogate Measures of Cell Mixture Distribution. And actually, this approach is really quite simple. It involves only 2 steps. The first step is we need to take our cell-specific methylation data, and we need to identify the CPGs that uniquely discriminate each one of the cell types. All right. So once we've identified the so-called fingerprint, methylation fingerprint of each one of the cells, we're then going to take those CPGs and we're going to go back to that equation that I showed you, and we're going to estimate the W, the weights or the proportions of the different cell types using constrained projection quadratic programming. And that's really it. Only these 2 steps. So this was really a landmark paper, and this method has since been dubbed the Hausman's method. And that's when you kind of know that you've made it as a mathematician or statistician, when you have a method that's named after you. So Andy's method really changed the game with regard to deconvolution. So here is a set of results from the original paper. They conducted an experiment where they purchased cell types and mixed them in predetermined proportions. So there were 12 different samples, and those 12 different samples are represented by columns in this data set. There were 5 different cell types that were used to create mixtures, represented by the rows. And so in the very first mixture, 90% of the mixture was monocytes, and only 10% was B cells, and there were 0% NK, T cell, and granulocytes, and so on and so forth. Second sample, we have 80% monocyte, 20% B cells, and so on and so forth. So these are the known mixtures, and this is what was predicted using DNA methylation. So what we can see is that doing DNA methylation-based deconvolution provides very, very close estimates to what the true known mixtures actually are. So this was really great and very encouraging. However, this was a very specific experiment. This experiment, we created artificial mixtures. So at the time, it was really unknown how deconvolution would fare in kind of a real, like a real data set. So around 2012, Lam et al. Published a paper where they had profiled methylation using the 27K array in 94 individuals. And this was methylation profiled in peripheral blood mononuclear cells. And in the very same samples that were used for methylation profiling, they had also collected CBC measurements. So this provided a really nice data set to see how well deconvolution performs as compared to kind of a gold standard measure of cell proportion, in this case, CBC. So when we applied deconvolution to this data set, on average, what we saw was really good consistency. So monocytes, whoops, sorry about that. So the monocytes and lymphocytes were predicted to be very, very close on average compared to their CBC measure valued. However, when we actually looked at the performance of monocytes and lymphocytes in the form of like a scatter plot and computed the correlation, we saw that there's a lot of room for improvement. So clearly, correlation of 0.6 and 0.61 indicates moderate correlation, but there's definitely room for improvement here. So if we go back to Andy's method, as I mentioned, it's pretty simple, just 2 steps. And when we think about which one of these steps would be a good candidate to prioritize trying to make an improvement, it's really step 1. So in step 1, this is where we identify the specific CPGs that distinguish different white blood cell types. And step 2 is really just about a mathematical, constrained projection, quadratic programming. It's a mathematical construct. There's really not a whole lot to improve in that sense, but we can definitely make some improvements in terms of what CPGs we select to use for deconvolution. And so in Andy's original method, he proposed identifying the CPGs using an approach that was based on an ANOVA model. So the thing about this ANOVA-based approach is that what you wind up getting, or what you can get, is CPGs that well discriminate certain sets of cell types from others, but don't do very well at discriminating all possible cell types. And so we see that here in this heat map. So in this heat map, our lymphocytic subtypes, CD8, CD4 T cells, and NK, are really well discriminated from our myeloid-derived cell types, our granulocytes and monocytes. But individually, these lymphocytic cell types, CD4, CD8, and NK, are not very well discriminated from one another. And so that's going to impair the performance of deconvolution to accurately predict the proportion of those cell types because we just simply don't have signal for distinguishing them. So a better approach would be to compare each cell type to every other cell type and rank order the CPGs that are differentially methylated in that cell type compared to all others. So when we do that, and we reconstruct the heat map, we now see that each one of these cell types at least exhibits some signal that distinguishes it from every other cell type. So now the CD4, the CD8, and the NK actually have some signal that distinguishes them from the other cell types. So if we look at the performance of these 2 different approaches, so we have ANOVA and we have the estimate cell counts, and there's 2 different ways that we can assess performance. We can compute what's called the R-squared, or the coefficient of determination. And we can also compute what's called the root mean squared error. So we want the R-squared to be high. We want that to be close to 1. That indicates good performance. And we want the RMSE to be low. That indicates good performance. What we see is that on average, the pairwise ranking of CPGs does a lot better than the ANOVA-based approach, not surprisingly for the reasons I just pointed out. We see that both with respect to R-squared and RMSE. So estimate cell counts, definitely improvement on top of ANOVA, but there's still room for further improvement. 0.76 is pretty darn good, but R squared, as I said, we want to get that as close to 1 as possible. So this leads us to a question about optimality. We believe that there is some optimal set of CPGs that when we have that set of CPGs, we can get really accurate deconvolution estimates. And so the way in which we can try to find that optimal set can be likened to a football example. So there are 53 players on an NFL roster, and there's only 11 players on the field at any given time. So for the purposes of this explanation, let's totally forget about different position groups, forget about defense, forget about offense and special teams. If we were to take 53, choose 11, there are a lot of possible combinations that we would need to go through to find the 11 optimal players that we should put on the field, right? The same is true for methylation data, except at an entirely larger scale. We cannot possibly go through all of these combinations. So is there a smarter way that we can try to find the optimal set? And the answer to that question is yes. So the way that this process works is that we begin by randomly selecting 11 players. Okay? And then what we do is we take one of those 11 players at a time and we evaluate the team performance with and without that player. So if we take Josh Allen and he is our player, we assess the performance with Josh Allen and we assess the performance without Josh Allen. If the team performs better with Josh Allen, then in subsequent iterations of our algorithm, we want to upweight the probability that Josh Allen is selected to be part of our set. If the team performs worse with Josh Allen, without Josh Allen, then we want to downweight the probability that Josh Allen is selected in subsequent iterations. And so that whole process in doing this for optimizing libraries for deconvolution was described in a paper that we published in 2016. And so I'm not going to get into the gory details of this algorithm, but there were a number of different contributions that we made. The first is how to assess prediction performance when we're doing deconvolution. We can use the R squared, RMSE, and in our paper, we used a combination of both. We also had to determine how to assess the contribution of each specific CPG as they relate to prediction performance. And then also how to update the selection probabilities at each iteration of the algorithm. So all of these questions were answered in our paper in a method called IDLE, Identifying Optimal DNA Methylation Libraries. And what we see when we implement IDLE is when we begin with a candidate set of CPGs, all of those CPGs have an equal probability of being selected to be part of the optimal library. However, as we move through the iterations of the algorithm, we see that the probabilities of some of those CPGs go up or go down depending on how well that CPG was determined to contribute to good prediction performance. And so what we end up with at the end of the day is a set of CPGs that have a high probability of being selected. And as we look at our performance measures as a function of the iterations of our algorithm, we see that as we move through the iterations of our algorithm, our root mean squared error goes down, which is a good thing. And our coefficient of determination, our R squared, is going up, which is a good thing as well. So to actually test this methodology out, we had a training and a validation data set. Our training data set consisted of whole blood samples on 6 adults, where we also had, in addition to methylation data, we had cell proportions that were measured using flow cytometry. So we had a measure of the ground truth. And then we had 2 different validation sets, both which were created using a reconstruction experiment where we purchased cells and we mixed the DNA in predetermined proportions. So using our training data, that was used solely for the purposes of identifying an optimal library of CPGs for deconvolution. And then using that optimal library, we applied it to our validation data sets. So the 2 different validation data sets are represented by squares and circles in this plot. And what we see is for these 6 cell types, we see remarkably good performance for deconvolution. So we see R squareds that are close to 1, we see root mean squared errors that are low, and we see minimal bias of our estimates. So most of them are close to or hovering around the dotted line at unity. So deconvolution is doing extraordinarily well here. In some later work, 2018 and 22, we have expanded the repertoire of cell types and also optimized deconvolution libraries for new Illumina arrays. So um, the results that I just presented were developed using the Illumina 450K array, but shortly after the 450K array came the EPIC V1 array. And so we published a paper in 2018 where we determined an idle optimized library for that specific array. And much like the performance that we got with the 450K, performance with the EPIC was really strong. We see R squareds that are close to 1 and really small root mean squared errors. In 2020, we extended the library to include 6 additional cell types. So going from 6 all the way up to 12 cell types. So we've added basophils, B memory cells, B naive cells, also CD4 memory, CD4 naive, CD8 memory, CD8 naive. And across these 12 cell types, much like the earlier performance that we saw with the 450K array and the EPIC V1, we see really strong performance of deconvolution. So there are some persisting challenges, and around 2022, I worked with some of my graduate students to try to address these challenges. So one of the persisting challenges around 2022 was that strategies like IDLE require a training data set with gold standard measurements. And so as we continue to expand the repertoire of different cell types that we want to deconvolve, we're not always going to have such a training data set. And so we wouldn't be able to use IDLE. And so one of my students conducted some work to try to tackle that problem. How do we select libraries when we don't have a training data set and we can't use IDLE? The other question that we need to consider is also limits of detection. So when we have any type of device for conducting measurements of something, it is wise to know what the limits of detection are of that technology. And so one of my graduate students published a paper where we took a look at how do we compute the limits of detection and uncertainty associated with methylation-based deconvolution. And then finally, an enduring challenge is proportion data are what we call compositional. They have a unit sum constraint. They all sum to 1. And as a consequence of that, there is a guaranteed correlation between the different elements of a composition. And that creates challenges when we analyze such data. And there are a class of statistical methods called compositional analysis methods that one of my students has been exploring over the last couple of years. So to talk a little bit about the compositionality, and this is kind of where I'm going to kind of wrap up the presentation, there are different things that we can do with cell composition estimates. One thing that we might want to do is to get those estimates and include them in our statistical models to adjust for the potential confounding effects. In fact, that's where all of this had its birth. We want to control for the potential for confounding. However, the estimates of the different cell types might be of interest in their own right. So we may want to look at the relationship between the abundance of different cell types and characteristics that we have on our samples. So treatment response or cancer risk and survival. So that's what leads us to what's called compositional data analysis. And to try to help you understand the implications of the unit sum constraint, I have a pretty simple example here. So we have 2 different patients, patient 1 and patient 2. And as we can see in this plot, when we look at the cell counts of 5 different cell types, we see that the only cell type where the count actually differs between patient 1 and 2 is neutrophils. All right. So if we were to conduct an analysis on the cell counts, we would find that only neutrophils should reveal a difference between patient 1 and patient 2. However, if we calculate proportions on the basis of the counts, where the proportions are calculated as the count of each cell type divided by the total for that patient, what we now see is that the proportions of lymphocytes, neutrophils, monocytes, eosinophils, and basophils look different between patient 1 and patient 2. And this isn't a surprise. The denominator of the ratio for the proportions we're calculating is different between patient 1 and patient 2. Therefore, these proportions are going to be different. This provides the illusion that the count of the cell types might be different between patient 1 and patient 2. So this is something that we need to account for, and the way in which we account for is through compositional analysis. So one of the papers that Alex published was a very, very simple paper. It was to simply take a methodology that was developed in the context of microbiome data, which shares similar compositional problems to cell proportion data. And this method is called ANCOM. And it was to take that method and actually apply it to cell proportion data and to see how it performs compared to kind of the standard approach that's used in the literature, where we're just fitting regression models independently to the proportions of each cell types. So Alex did this in his paper, and what we found was if we look at the false discovery rate, now we want that to be low, ideally controlled at around 0.5. The standard approach, as we increase the sample size of our study, that false discovery rate absolutely blows up. It becomes uncomfortably large very, very quickly, whereas ANCOM is well controlled down near zero. In terms of statistical power, which is the probability of detecting a difference if it really exists, we see very similar performance between ANCOM and the standard approach. So similar power between ANCOM and the standard approach, however, much better operating potential when it comes to false discovery rate for ANCOM. So this was a really nice paper that we tried to get out this information to the epigenomics community, analyzing deconvolution estimates. And Alex has gone on to conduct some other work for compositional analysis for deconvolution estimates. And in the interest of time, I'm going to skip past this. And I'm just going to bring you to the end here, which is kind of where we currently sit. So this is a chronology of deconvolution, kind of where we started back in 2009 and to where we are right now. So there are a lot of open questions. I think that some of the open things that people are working on is continuing to expand the repertoire of cell types that we're actually capable of deconvolving, and to actually utilize this idea in different types of tissues. So deconvolution really had its origin with deconvolving white blood cell, whole blood, but we can apply the same type of idea to other tissue types. We also want to be mindful of forward compatibility. So every couple of years, Illumina introduces a new array, and we want to make sure that our efforts in developing strategies for deconvolution on earlier generations of the arrays have forward compatibility with some of the new generations that are forthcoming. So the vision that I have for the future and working in this area is we want to be able to scale DNA methylation-based deconvolution and be able to actually utilize this for clinical applications. We feel that, at least in compared to flow cytometry, this can be a lower cost solution. In certain scenarios, it can be more efficient. And there are a number of different practical advantages DNA methylation-based deconvolution has over current cell surface-based cytometry methods. So some important lessons that I've learned along the way, and this is what I'll leave you with, is being a student of the scientific field that you work in. So this is more directed for anyone on this call that's more quantitatively or data science oriented. We are often working on a lot of different types of projects, and I think it's important for us to really become stewards of specific areas and learn the science of whatever the main areas we're collaborating in. I think it makes us stronger collaborators, and at the very least, at least we will have a common language that we can share with our collaborators. Another thing that I'd like to share is, sometimes the solution goes beyond the specific statistical method. So there was really no change that we made to Andy Hausman's deconvolution method. The contribution that was made that really elevated and took deconvolution estimates to a whole nother level of accuracy was just changing around what CPG sites were used to build the library. It had nothing to do with the statistical method at all, just what the substrate was that was being inputted into the statistical method. That's what changed. I also feel like being open-minded and also open to criticism and critiques of our work. And then finally, one thing that I have learned over the history of the deconvolution methylation work that I've done is the value of teamwork. And that's where I'll finish. This is the team that I have the great privilege and honor of working with. The folks on the left-hand side are members of my lab. And then the DNA methylation cytometry team, there's Andy Hausman, who pioneered the Hausman method, and then Carl Kelsey, John Winke, Brock, Lucas, and Annette are longtime collaborators of mine. So I really appreciate your time. I would be happy to take any questions with the remaining time. Thank you very much.
Dr. Vivek has a question.
Sorry, I don't have any specific question. I was just clapping. Thanks.
Thanks. Dr.
Hey,
Chris?
Amy.
It's a wonderful presentation, and our team is actually working around this topic, so it's very interesting to hear your thoughts.
Thank you.
Hey, Devin, this is Andy Nelson. Can you hear me?
Yeah,
Sorry,
I can
I'm
hear you
on
just
my cell phone.
fine.
Hey, thank you. This is a great presentation. I agree. And I'm sorry if I didn't necessarily, if you said it explicitly, but as you think about forward compatibility and as you think about, different, spectrums of arrays coming out in terms of number of sites, do you think that the more sites we measure ultimately generates better data, and should we really be thinking about as whole genome sequencing costs plummet, and as methyl-seq becomes whole genome methyl-seq becomes a real cost-effective possibility? Do you think that might be the next emerging way to even push further forward with deconvolution and assessment of methylation patterns in different types of samples?
I think you raise a really good question, Andrew. Truth is, I'm not totally sure, but let me tell you what I do know and what I've seen. So, when we started this deconvolution adventure, this was when we had the 27K array, right? The Illumina 27K array. So this was back in like 2010, 2011. And the performance, now granted, at that time, there were only 6 cell types that we had purified methylation signatures on that we could actually use for deconvolution, right? It was CD4 T cells, CD8 T cells, natural killer cells, B cells, monocytes, and granulocytes. So from the 27K to the 450K array, there was definitely an improvement in the performance, in the accuracy of the estimates that we were getting. Perhaps not surprisingly, the number of CPGs going from 27K to the 450K array is a pretty significant difference. So you might expect there to be an improvement there. Now, from 450K to 850 to the EPIC array, which has about twice the number of CPGs, there wasn't that big of a jump in the performance in terms of improvement, because the ceiling that we could improve upon where we were with the 450K array was already really high. Does that make sense? Like, we were already really getting good performance with the 450K array, just having those 450,000 CPGs. So the margin for improving was really limited. Okay. Now, where I think that there's value in increasing the resolution is not necessarily deconvolving the cell types that are already part of the libraries that we have, but deconvolving the ones that we have yet to profile. So there are certain other types of exotic cell types or cell states where it becomes really, really difficult, like, to identify CPGs that uniquely discriminate those cell types from every other cell type. So, like, if we think about like myeloid-derived suppressor cells and granulocytic-derived suppressor MDSCs, it gets really difficult to deconvolve those cell types. And so I think that there is potential, as we get more resolution, we get more coverage, to be able to find the signal that discriminates those cells. So that's where I'm hopeful, and why I still think it's a worthwhile endeavor to continue doing this. I don't know if that helps provide some clarity to your question. Maybe I went a little bit of a tangent, but
no, I think that's great, actually, and I think your point on the cell states is super insightful, and obviously, we're always kind of dealing with law of diminishing returns in terms of, should we keep measuring more things, or do we do it with spatial, or how many different things can we measure on a single cell analysis? Those types of questions. But yeah, I agree from a cell state perspective, and trying to apply it to different forms of biology. I really think that was an insightful answer. So super appreciate it.
Thank you. Shannon?
Hi, Devin. Shannon
Hi.
Sullivan. I'm an epidemiologist. I've been working for the past year with DNA methylation data, so this is a new area for me. And I have 2 questions. How well does your IDOL method work with the EPIC version 2? And then I'm sure everybody has asked you this. Are you working on updating your IDOL method to account for the additional CPGs with the version 2?
Yeah, that's a great point. So we have not yet updated the IDOL method for the EPIC v2. So that's still something that's ongoing. How do I expect it will perform? I mean, I think it will perform well, and I know that sounds a little bit self-serving, but let me kind of explain, rationalize why I think it will work well. So there is a lot of similarity between the EPIC v1 and the EPIC v2, right? There are some things that make the v2 different, but there's a fair amount of overlap and similarity. And what we know from the v1 EPIC array is for the 12 cell types that now belong to the extended library and the Nature Communications paper that was published in 2022, there's outstanding performance that we see for those 12 cell types. R-squareds are all 0.95 and above. So I think if I'm just extrapolating the performance we see with the V1 to the V2, I think it will still be really, really strong. It's hard to say, though, how that might be impacted by introducing new cell types to the library, whether or not the EPIC v2 array, the differences in the CPGs being profiled between the V1 and the V2, if there's additional signal in there for discriminating new cell types that you add to the library, that's where it becomes challenging for me to make a judgment call or assessment on how good the performance will be. But we are actively revising the IDOL library using the V2. That's ongoing work that Brock and Lucas, my collaborators at Dartmouth, are working on with some of their students. But we haven't yet finished that work. So the jury's still out.
Thank you.
Sure.
Anna, go ahead.
Yeah, so there were good questions and smart questions. Now I have a stupid question. For instance, we are studying epigenetics, methylation with some outcome. And then we need to adjust to account for cell composition, as we talk about it. So is it okay to use the same data to create cell composition to adjust for? So using methylation and immune cells from the same data.
Yeah, so if I'm understanding your question, let's take a step back. So if we were going to adjust for some other confounder, let's say some type of clinical variable, and supposing that we had a measurement of it, we would just put that in our statistical model, and that's how we would control for the potential confounding effects. Your concern is if we are interested in studying DNA methylation, say we want to compare CPG-specific methylation between diseased and non-diseased, and we know that cell composition can be a potential confounder, and so we want to adjust for it in our model. So the concern is we're using methylation to estimate the thing that we are then going to include in our statistical model. Is that, am I understanding that correctly?
Yeah, thank you for explaining my question in such a great detail. Yes, it's absolutely correct. Yeah.
So you're right, but I think you're right to have this concern and this question, but what we are doing with the methylation data is we are transforming it in a way to get actual proportion estimates of the different cell types. All right. Yeah. So we're not actually adjusting for the methylation data per se. We're taking the methylation data through a series of steps and using that to estimate some entirely new quantity. So from a statistical standpoint, I don't see any problems with the fact that we've used the data that we're interested in fitting a model to predict something that we're adjusting for in our model. I don't see that as an inherent problem. In fact, there are some other reference-free methods for adjusting for cell composition, like remove unwanted variation, RUV, or surrogate variable analysis, that essentially do the same thing. And if you think back even to genetic data, where you control for population stratification by computing principal components on the genetic data, you know, your SNP data, and you include those PCAs in your model, you're kind of doing the same thing. So I think it's okay, but I understand why the question would be raised.
Yeah, thank you.
Yep, no problem.
And the other question is, you develop more, try to study more immune cells, and at the same time, flow cytometry also try to measure more cells.
Yes.
So do you think your method, how fast are both methods developing in terms of measuring, getting to new cells? It seems like epigenetics could be a little bit faster, but I don't know. Yes.
Well, it kind of, epigenetics depends on flow, right? Like we need to get isolated cell types from somewhere.
Yeah, so
we can't, naturally, I think epigenetics has to go at a pace that's slightly slower than flow because we depend on the cells isolated from flow to actually be able to get their signature. If that makes any sense.
Yes, and flow has, I see. So it's the only method that that allows you to develop. Okay, that's another stupid question, but that's good to know the answer. Thank you.
No
Very
problem.
helpful. And great presentation. Yes, I appreciate you coming here.
Thank you.